It's wrong every time. Didn't you think it was due a different time too? What you would have learned after the first question.
But what? Definitely.
You know, my mind's kind of shut off.
I know, see, this is what I'm trying to deduce. Scientific methods, then, if I come over here and you all migrate back.
Not that lag, but that's
Okay,
You'll make measure
so by and large, we've clicked in. And if you haven't, by the way, class has started, click in. And we've largely got this idea. Yay! Again, ion exchange is all about what are the charges on our molecule? What are our ionic states? But just as a quick reminder to ourselves,
I feel like that happens a lot here.
You just
I
gotta
keep
pick
putting
up
them
the...
back up there.
Someone must
You
have
have to keep throwing them away.
Or someone doesn't like you and they know that you're in here, so they keep replacing with crappy ones.
full story, one time I threw one and it hit someone right in the face. I felt really bad.
Did they come to school the next time with like a full face helmet on? Just in case.
Well, we didn't give it into the part. Typically, I break one of these in half every semester.
Wait, how, like pointing or like?
I'm doing a
like
demo.
leaning? Oh.
Anyway, if we have beads that have positive charges, which allows some proteins that are, say, negative charged to associate, whereas proteins that are positive charged don't associate, what would we call this type of column? So it's positive
Affinity?
beads, positive proteins then will be repelled, negative proteins will stick. This would be an anion exchange. Okay, so that's a big thing, just that it's named after the proteins that stick, not the bead charge. It's one of those things that I just want to highlight and make sure we feel good about. But here's the deal. Again, we're now far enough into the class that I want us to start being ready for all the things that are coming, like ACS finals and MCATs and GREs. And my multiple choice questions and the quizzes, if you haven't noticed, are written very technically. I'm trying to prepare you for those kind of things. But we're going to get more clicker questions that drive in that direction as well, and that might be harder or use more technical terminology to try to really reinforce that. So this next question that I've now built up, and you're going to be like, that's not hard, or maybe it will be, I really encourage you, talk to a neighbor, work through it, talk about how you work through it, make sure you're on the same page. So here's a harder question about the stuff we talked about on Monday. Check it out. All right, don't forget to click in once you've discussed it with your neighbor. Not to rush your discussion, but make sure you're discussing.
Has to do with sizes. And then the gel's charged, so it's in the combination. Yep.
Here's what I live for as a professor, your pain. No, there's two answers that people both like a lot. So let's talk about it. A and D seem to be the front runners. So I'm going to hide this so we can actually read it. But I'm keeping it open, so if you change your mind, you can change your vote.
Because it changes our score.
What?
Because it changes our score.
Because it changes your score so much. But it might make you feel better inside. So let's first talk about why B and C aren't there, because we're A or D. So let's talk about B. Involves the disruption of quaternary structure, but not tertiary structure. Let's break it down. Disrupting structure at all. Why would we think there might be structural disruption? SDS gets rid of structure. Okay, but so why isn't B true? Yeah, it disrupts tertiary as well. It disrupts everything. Absolutely. So it's not B because not only will it disrupt that, but it will also disrupt that. So this not is lying to you, right? So good. C, achieves separation based on the isoelectric points. Why not? Ooh, I like that, Marlia. SDS makes the entire strand negative. So what? That's not an isoelectric point. That's true, you're right, but let's say more. Like, why would isoelectric points not matter anymore? Explicitly. Maybe I'm being dense. Let's say this. So if I have a native protein, and it happens to have some positive and some negative charges based on its isoelectric point, so that its net charge at our thing is +3, seems to be based on isoelectric point, whether it's positive or negative. So why does SDS make that not matter?
Changes the structure.
It's exactly what Marlia said. It just adds a metric boatload, avoiding cursing, of negative charges. It doesn't matter if this thing had a charge of +3, when after you add SDS, the net charge is like -250. Negative 247 versus negative 250 is negligible. It's lost in the wash. So the ionic state and the native charges of the protein stop mattering. Cool. Okay. So now we're down to A or D, which have to be true. So let's start with it. A, involves protein detergent complexes. I like it so far. We're analyzing proteins, and SDS is a detergent. I like it. I like it. That vary in charge to mass ratio. I don't know. By the way, I'm doing this explicitly, test-taking strategies. What about when you get to something you don't know? I don't remember talking about charge to mass ratios with proteins. Ignore it. Just ignore it. It involves protein detergent complexes. Cool. That don't vary in mass. They do vary in mass. We have big proteins. We have small proteins. We separate them by their size. And in fact, if we did think more about it, so let's imagine we have a small protein and a big protein, and we treat both with SDS. This one will unfold, and it'll get negatives along its whole length. And this one will unfold, and it'll get negatives along its whole length. So they're different in size, but note, for any given unit of mass, it has the same charge density. So in fact, they don't vary in charge to mass ratio. They have the same charge to mass ratio throughout, but they vary in mass. So let's look at D then, which I'm leading you towards. Yields the subunit molecular weights. I have no idea. Cross it out. Yields molecular weights. I don't, subunit, whatever. Yields molecular weights. Yeah, we use this technique to look at molecular weights for an oligomeric protein. I have no idea. Cross it out. One of the dangers of multiple choice questions is this. Think like a test writer for a moment. Usually if you write the right answer, it's pretty obvious. Like what technique separates, what column separates proteins by size? Correct answer, size exclusion. It's the only one with the word size in its name. It seems too easy. So test writers, make the correct answer tricky, because they don't want to write it just like easy every time. So it's often easiest to find the wrong answers, and then the right answer has all these words you don't know and all this stuff, but you're like, whatever, the other ones are wrong. There we go. And I think that's a really strong strategy. Ignore what you don't know, try to solve it based on the knowledge you have. So let's talk about adding a little knowledge here. If instead of a small protein or a large protein, we had a protein that had quaternary structure, it was actually a trimeric protein. When we treat it with SDS, what would we see? 3 different strands. They'd all move at different rates based on their size. So yields the subunit, not the whole thing, but the subunit molecular weights for an oligomeric protein. Here's where a little Greek helps. Mer means thing, oligo means few. It's got few pieces. An oligarchy is ruled by the few. So, oligo, few. You see that in DNA all the time. We ordered an oligomer. What? You mean a polymer? No, just an oligomer. Where's the line? I want to be Jerry Seinfeld. This kind of question, this one was pulled straight out of GRE. So the biochemistry GRE, I pulled this straight out of. Can come out of. So where we're at. Do we feel okay about protein purification stuff that we talked about last time? Are there lingering like, hey, I don't actually get this piece? Yeah, hit me.
Would you ever do just one column chromatography, or would you usually do ion and size exclusion? Yeah. And I'm sure there's more than just the 3 we learned too.
Oh yeah. So in reality, in real life, you typically do at least 2 to clean things up, but not always. So this is a great opportunity. I mentioned that affinity columns can be very good. So hopefully we remember affinity columns. This is a great question from Marley, so let's talk about it. Affinity columns rely on the fact that your protein has some unique function. But the limitation is that it probably doesn't have a unique function. What I mean by that is, if I want to purify a DNA binding protein that binds DNA, I could have a bead with DNA on it. But we know that in a cell, there's all sorts of transcription factors and RNA polymerase and DNA polymerase. There's a lot of proteins that bind DNA. So this is specific, but not specific enough. So about 20 years ago, some very smart guys, I think the first people to ever do this were actually at Cold Spring Harbor out east, said, well, if our protein doesn't have a unique function, that is, it has a function, but so do other proteins. Let's give it a function that doesn't exist in nature. And what they noticed is that essentially, asterisk, no natural proteins bind to nickel. Like if you have metallic nickel, no proteins stick to it. In fact, that's why it can be a potent allergen. There's nothing on your skin that can like bind to and neutralize it. There's not really anything that occurs in natural biology that binds to nickel very well. And so they said, let's take our protein, whatever it is, and let's genetically engineer it to have a nickel binding domain. Make a new function that doesn't occur in nature, and then use beads that are coated with nickel. Potentially, if we're smart and things work like we want, our new genetically modified protein, our protein that has this new functionality in addition to its normal stuff, will bind and nothing else will. This idea of attaching this novel functionality is typically called tagging the protein. And there's a variety of these novel functions that we can introduce. This one, the nickel binding tag, is commonly known as a his tag, because it turns out to get a nickel binding domain, all you need is like 6 to 8 histidines in a row. Like literally, if we look at the structure of this nickel binding domain, we zoom in, it's not a good zoom arrow. If we zoom in to see what is this, all it is histidine, 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 histidine. They form a loop and they bind nickel inside. For whatever reason, evolution's a roll of the dice, there's not really any natural time when 7 histidines occur in a row. It just hasn't happened evolutionarily. And so by introducing that new functionality, we can get really specific binding. And in fact, it lets you get really lazy. In my actual research lab, the majority of our proteins, we do just one column. We do a nickel column with our his-tagged proteins, and they come out like 99% pure. And that last 1% went away. Good enough for government work. So the his tag is a very common one. There's other ones like the flag tag or the GST tag. But this requires you to go back and do your cloning, right? This requires you to say, hey, when I make my plasmid, I'm going to add on this piece that codes for this his tag. And in reality, this is so common. When you go to buy a plasmid, they all have these tags built in. They know you want to tag your protein, so they just have a tag in there ready to go already. So that works. Yeah, hopefully that's something. His tags are cool. Any other questions about protein purification stuff? That was good. Reminders. All right, we're going to move on. Fools, you could have wasted the whole class. So last time we talked a bit about the idea of protein denaturing and how we can relate that to the concept of cooking or baking, that proteins losing their structure cause physical changes that we can measure and care about. The negative side of that, so now we get to be very serious and sad, is when things start denaturing inside your body, it can cause severe disease states. And I want to bring up 3 examples of when protein folding goes wrong to various levels of detail. And the first one I want to start with is Alzheimer's disease. There's some caveats that need to be given out here. So this is, I don't know why I don't actually have that word on the slide, so let's fix this. This slide is about Alzheimer's disease. And the challenge of Alzheimer's and talking about it in an academic setting is we don't have all the answers yet. We'd like to present things we can tell you from beginning to end how it works. And we don't know that for Alzheimer's disease. We do know that it's a disease that has clearly multiple causal factors, it has multiple symptomatic factors, and it causes all sorts of different bodily harm. So I'm just going to talk about one facet of Alzheimer's. Just one facet. There's at least 2 major cellular level mechanisms at work with Alzheimer's, and I just want to talk about one of them. So here's the idea. There is a protein that naturally exists in the membrane of your nerve cells. So it's produced by your body, inserted into the membranes, and serves some kind of important function. Exactly what it does is not fully known. It has to do something with successfully forming synapses, so interactions between nerves, and ion import and export, so allowing chlorine and potassium in and out of the cell. But the exact role, why it's needed, we don't really know. But we know that it is necessary. That is, if we breed mice where we've genetically deleted the gene for this protein, they don't survive the 4-cell stage. Like, it's deadly. You have to have this protein to be alive. By the way, that's like a lot of proteins. We know you need it. We don't quite know why. Under some conditions, one of the domains of this protein gets cleaved off from the rest of the protein. And that's thought to be natural. We actually have enzymes that do this process, and it's thought to be involved in neural signaling. All natural so far. Here's where it goes wrong. This protein has, this piece of the protein that gets cleaved off, has a folded structure. It has a native structure that it should be in, and presumably that structure is involved in cell-to-cell signaling or synapse formation or something. We conserved it in evolution. If you delete it, it's embryonic lethal. It's doing something important. We don't quite know what. But it's necessary. But under some circumstances, it partially